Applications of recombinant DNA technology. The biotechnology in the early 1970s was concerned with the generation of commercial products through the natural metabolic action of microorganisms. However, the nature of biotechnology was changed forever with the advent of recombinant DNA technology. This technology allows the construction of desired recombinant DNA molecule at will. The production of human insulin in bacteria, which seemed like a science fiction some few decades ago, was made possible due to development of recombinant DNA technology. The laborious and time-consuming conventional methods of developing new plant varieties can be replaced by genetic engineering techniques to produce transgenic plants of desired characters. Today, let us discuss some of the important applications of recombinant DNA technology in the field of agriculture and medicine. Herbicide-resistant transgenic plants. There is about 10% loss in global crop production annually due to weed infestation. The unwanted weeds have to be removed using herbicides, but these chemicals do not discriminate weeds from useful plants. The transgenic herbicide-resistant plants developed through genetic engineering techniques can withstand deadly effect of herbicide, thereby allowing only the destruction of unwanted weeds. Glyphosate is one of the commonly used herbicides which inhibits the enzyme 5-enol pyrovyl schemate 3-phosphate synthase, in short EPS-PS, which is involved in aromatic amino acid synthesis in plants. Three strategies are usually employed for production of glyphosate-resistant transgenic plants. First one, overproduction of EPS-PS enzyme. Secondly, production of EPS-EPS enzyme not affected by glyphosate. Third, production of COX enzyme which deactivates the glyphosate. The mutant EPS-PS gene and GOX have been used for the production of Roundup Ready maize variety by Monsanto, USA. Now let us take up insect-resistant transgenic plants. Insect-resistant transgenic plants are produced through genetic engineering of crop plants with insecticidal toxins. One of the insecticidal proteins is the crystallized or cry proteins produced by Bacillus thuringiensis, which kills harmful insect and larvae. The cry proteins are produced by chrysins. The protoxin cry protein, having the size of 130 kilodalton, has toxic function localized in N-terminal half, while the crystalline nature is produced by C-terminal half. When the cry proteins are ingested by the insects, proteolytic cleavage is produced in the alkaline environment of insect midgut, producing a 60 kilodalton toxic core fragment. The cry proteins are about 80,000 times more toxic to the target insects than those conventionally used organophosphate insecticides, and also are highly selective in their actions. Insecticidal protein genes such as Cry1AA, Cry1AB, and Cry1AC derived from Bacillus thuringiensis subspecies Karstiki have been used to develop insect-resistant transgenic plants. The low level of chrysin expression can be improved with the use of truncated zinc containing only the N-terminal portion and a strong promoter. The commercially available Bt cotton is developed with Cry1AC protein from Bacillus thuringiensis and is resistant against insect Helicobarba amergira. Next, slow ripening fruits. Antisense RNA technology is used to develop transgenic tomato with slow ripening fruits. The antisense gene of polygalactoronase enzyme is introduced into the plant, which also contains the endogenous normal polygalactoronase gene. The transcription of the two genes in the nucleus yields antisense and sense RNA transcripts, which will best pair to produce double-stranded RNA molecule. This event prevents the translation of messenger RNA, and double-stranded RNA is destroyed by RNAs present in the nucleus. The transgenic tomato, developed by Calzin, a US-based biotechnology company using antisense RNA technology, is known as Flavor Saver tomato. The US Food and Drug Administration on 18 May 1994 ruled that the Flavor Saver tomato was as safe for human consumption as normal tomatoes that were produced by conventional means. Next, let us take up transgenic colon rice. Vitamin A deficiency is a common problem in those regions where rice is consumed as staple food. This is because the rice grain does not contain provitamin or beta-carotene, which is important for vitamin A production. Deficiency of vitamin A produces night blindness among the children. Transgenic rice can be created which contains high quantities of beta-carotene and are called golden rice because the grains are yellow or golden colored due to rich content of beta-carotene. Three transins which provide phytone synthase, phytone desaturase, zeta-carotene desaturase, and lycopene cyclase activities are transferred into rice using agrobacterium-mediated gene transfer. The transgenic golden rice contains provitamin as high as 85% of the total carotenoids present in the rice grains. Next, we have molecular farming. The plants are used as bioreactor for large-scale production of proteins and chemicals, which can be completely novel to them. The advantages of using plants as bioreactor are that they are easy to grow and can generate considerable biomass. In laboratory scale, plants have been used to produce bioplastic, polyhydroxybutyrate, monoclonal antibodies, vaccines, and a number of potential therapeutic agents. Let us take up the production of bioplastic in plant. The production of bioplastics or biodegradable plastics in plants is one of the important examples of molecular farming. Polyhydroxyalkanoids (PHA) from the bioplastics and polyhydroxybutyrate (PHB) is the best characterized PHA, which is usually found as intracellular inclusions in wide variety of bacteria. PHB accumulates as high molecular weight polymer in alkalogens eutropos up to 80% of the bacterial dry weight. Expression of PSA-A, PSA-B, and PSA-C is essential for polyhydroxybutyrate production. In one scheme of transgenic plant production containing PSB, only two genes, PSA-B and PSA-C, were introduced into Arabidopsis plants because acetoacetyl-CoA was already present in the cytoplasm at the start of the pathway of isoprenoid production. The microbodies of bioplastics were randomly formed in cytosol, nucleus, and vacuoles as the introduced two genes were not tagged with target peptide sequences. The amount of bioplastics was relatively low, 20 to 100 microgram per gram fresh weight. In the second method, all the three genes, PSA, PSA-B, and PSA-C, were transferred into Arabidopsis plant after tagging with Robisco's small subunit transit peptide. The PSB biosynthesis pathway occurred only in the chloroplast and PSB accumulation was more. Now let us take off application of recombinant DNA technology in medicine. Vaccine. Conventional vaccines contain either a kill inactivated or a live non-virulent attenuated form of an infectious agent capable of evoking an immune response. There are many limitations associated with present system of vaccine production. Some of the drawbacks are difficulty to grow infectious agent in culture, the use of animal cell culture to produce animal and human viruses is expensive, possibility of reversion of attenuated strains to virulent form, and limited shelf life of the vaccine, and requirement of cold fridge for storage. With the advent of recombinant DNA technology, there are possibilities of generating new vaccines which can overcome the shortcomings of traditional vaccines. The reversion of attenuated infectious agent to virulent form can be prevented by generating a genetic engineered agent which could be used as a live vaccine. In this case, the virulent genes can be deleted from the infectious agent, but still it can stimulate an immunological response. The infectious agent will not be able to reacquire the whole deleted virulent genes while growing in pure culture. In case of infectious agent which cannot be grown in the culture, the genes with codes for proteins having critical antigenic determinants can be isolated, cloned, and expressed in another host like E. coli. The proteins derived from the new host can be utilized to develop vaccines. Next is the identification of disease genes. The development of recombinant DNA technology has revolutionized the search and successful identification of many defective genes which cause a number of genetic diseases like Huntington's disease, cystic fibrosis, Tay-Sachs disease, Howe-River syndrome disease, etc. Huntington disease (HD) is progressive neurodegenerative disease caused by an autosomal dominant mutation. The Huntington gene is located at the end of short arm of chromosome 4 and spans about 210 kilobase. A normal Huntington gene contains a trinucleotide repeat (CHG) of less than 36 copies, while the mutated gene comprises more than 36 copies up to 100 of the trinucleotide repeat. A simple and accurate DNA test for ISD mutation can be provided using recombinant DNA techniques. If the nucleotide sequences of Huntington gene on either side of the trinucleotide repeat region are known, oligonucleotide primers can be prepared and used to amplify the region by PCR. The number of CSG repeats could be determined by polyacrylamide gel electrophoresis. Thus, individuals at risk for carrying the mutant Huntington gene can easily be tested for its presence. A test can be easily performed prenatally on fetal cells obtained by amniocentesis or cryonic biopsy, as the DNA required for performing the test is very small. The individual can be tested to check whether he or she is carrying the defective gene through the simple DNA test. Next, let us see the production of eukaryotic proteins in bacteria. Human insulin was the first human protein produced in microbes using recombinant DNA technology in 1982. Since then, several other human proteins like blood clotting factor 8, plasminogen activator, and human growth hormone have been produced in microbes at commercial scale. The human growth hormone is the second genetically engineered pharmaceutical protein to be approved for human use by US Food and Drug Administration after human insulin. This is a single polypeptide chain of 191 amino acid long and is required for normal growth. The human growth hormone coding sequence must be placed under the control of E. coli regulatory elements in order to express successfully in E. coli. The human growth hormone coding sequences was joined to the promoter and ribosome binding sequences of E. coli lac operon. To make this happen, the restriction enzymes ASAE3 cuts the cDNA of human growth hormone obtained from cDNA library of human pituitary gland at a site located of the 23rd codon of the gene. The sequence representing the first 23 codons of the gene was chemically synthesized and joined to partial cDNA sequence encoding amino acids 24 to 191. The whole unit was then inserted into the plasmid carrying the lac regulatory signals and introduced into E. coli by transformation. The human growth hormone produced in E. coli contained methionine at the amino terminus. The human growth hormone so synthesized in the E. coli was fully active in spite of the presence of extra amino acid in the form of methionine. Human gene therapy. Gene therapy offers the most promising approach to successful treatment of some of the inherited genetic diseases which are difficult to treat. Gene therapy involves adding a normal copy of the gene in order to replace the defective copy in affected individual. The gene which has been introduced into the individual is called the transgene, and the recipient organism carrying the introduced gene is termed as transgenic. The normal transgene will synthesize the missing gene product to restore the normal phenotype of the affected individual. The first gene therapy was performed on 4-year-old girl suffering from adenosine deaminase deficient severe combined immunodeficiency disease, in short ADA-SCID, in 1990. Individual with SCID lacks adenosine deaminase ADA, because of which there is accumulation of deoxyadenosine at toxic levels in T cells. There is no immune system in the SCID affected individuals due to the absence of T cells. The newborns with ADA SCID seldom live more than a few years. For the successful gene therapy of ADA SCID patient, a retroviral vector was first constructed with a human ADA gene under the control of strong promoter SB40. The white blood cells from the patient were isolated and the functional copies of the gene were introduced into the white cells with the help of retroviral vector. The ADA containing retroviral DNA was integrated into the chromosomes of the white blood cells. The transgene expression was verified by growing the transformed cells in the culture. The white blood cells with ADA gene were finally infused into the patient's circulatory system. The synthesis of ADA restored the immune function of the affected individual. The treatment must be repeated periodically to improve immune function of the patient. So, repeated infusions of the white blood cells with functional ADA genes have been performed on the patients. Finally, we'll come to the conclusion. Recombinant DNA technology has wide range of application in the field of agriculture and medicine. Several important agricultural crops have been transformed with transgenes to produce new varieties of transgenic plants with desirable characters. Plants have been genetically manipulated and engineered to be resistant against harmful insect pathogens, viruses, and herbicides. Transgenic plants with improved nutritional contents in terms of amino acids, fats, iron, and vitamins have also been successfully developed. Plants have also been genetically engineered to be used as bioreactor for large-scale production of therapeutic proteins. Production of new recombinant vaccines, therapeutic protein, identification of defective genes, use of DNA fingerprints in solving forensic cases, and treatment of rare genetic diseases with gene therapy are some of the important applications of recombinant DNA technology in medicine. With the present pace with which the molecular and genetic engineering research is undergoing, there is a high possibility that many recombinant DNA technology products may soon occupy a dominant position in the field of agriculture and medicine.